Okay, I guess we'll just get into it. I'm Lauren, I'm at the University of East Anglia, and I'm going to be talking about how your social circle can change how you age. So across the animal kingdom, males and females frequently differ in their lifespans and their rates of aging. So for example, in modern humans, women tend to outlive men. And this is a pattern that we can see across a range of different animal species, particularly mammals, where females will tend to outlive their male counterparts. And of course, there are examples of the opposite trend as well, where males outlive females. But although there exists this kind of diversity in rates of aging and lifespan, there still remains a general expectation that females will outlive males, and that males should essentially live fast and die young. And so one such reason we have this expectation is that there are sex differences in life history traits that are underpinned by anosogamy. And so that basically means that males who produce more numerous smaller gametes in the form of sperm tend to be limited by the number of available females. And so males are therefore expected to invest more of their energetic resources into gaining mating opportunities, which might mean that they will invest into fighting ability or weapons or even their sperm, which can come at the expense of investing into other traits like growth or somatic maintenance. And on the other hand, females are then expected to outlive males and basically live long and die old. And that's because females are often the sex that invests more into reproduction and parental care. So eggs are larger and costlier to produce, and so females often need to take time out of the mating pool in order to care for their offspring. So they can't really afford to live that risky lifestyle that's associated with males. And so going back to males, they tend to live risky lives in the pursuit of mates that can increase their mortality risks. So males that monopolize access to females can face frequent challenges from rivals that can increase their risk of injury and associated energetic costs of having to consistently win fights against other males. And these are all things that can accelerate their rate of aging. And so these so-called mating costs can occur due to trade-offs between the amount of energy that males can invest into lifespan relative to reproduction. And so males that invest more heavily into reproduction are therefore expected to suffer from reduced lifespan and faster aging. And that's all well and good, but we also know that males and females can influence the costs of reproduction for each other, particularly males influencing the costs of reproduction for females. And so males sexually harassing or transferring toxic sperm can increase the rate of reproduction of females, which can then make females age faster and have shorter lifespans. And so when we have this conflict over reproduction, it can also result in elevated mortality rates for females in their later life. And so males can accelerate female aging and decrease her lifespan through this kind of interaction. And what's more is that in nature, males and females don't just interact with each other, but they interact with members of the same sex, probably more often than they interact with the opposite sex. So how do these kinds of interactions influence the expression of life history traits in males and females? And this is where we still have kind of a limited understanding of how different socio-sexual interactions can influence life history traits in males and females simultaneously. So when we consider all of these in a broader picture, should we always expect males to live fast and die young? And so to see how robust these kinds of life history strategies are for males and females across these different socio-sexual environments, we decided to conduct this really big experiment to look at the life history traits of individual males and females across a broad range of social environments. And obviously we used Drosophila because that is the system where you can do this quite easily, and you only have to wait about 3 months for things to die, which is great for me. And so with this kind of experimental design, we expected to see strong and opposing effects of the social environment on key life history traits like aging, lifespan, and also fitness. And so to do this, we took individual males and females, and we randomly assigned them to one of these 4 social treatments for a period of 5 days. So we had flies that were either alone, with only the opposite sex, with only the same sex, or in equal sex ratio mixed sex groups. And so on day 6, after their first social experience, we then took our focal flies and we gave them, all of them, regardless of their social experience, a reproductive opportunity for 24 hours. So this meant that our focal males and females were given a single, young, virgin mating partner for 24 hours. And this is important because it allowed us to compare age-specific reproductive effort for each sex across all of these different social environments. And that's a really key difference between other studies because it's important to consider that flies will mate throughout their life. And this is one way in which that we can standardize that experience for flies. So during this 24-hour mating assay, we also looked at behavioral data. So we observed flies together for the first 2 hours that they were together. But I'm not going to talk about that data today for the interest of time. But after that mating experience, we then took our focal flies and we put them back into their social treatments with some new, young social partners. And this was repeated each week until flies died. And then we kept those mating vials to get a proxy of fitness for each of our focal flies. And so what did we find with this experimental design? So surprisingly, we found that males outlived females across all of those different social treatments. But how the social group was composed also influenced lifespan, and this was kind of in a sex-specific manner. So as expected, we found that both sexes had the longest lifespans when they were kept alone, and so that's the dark blue line. But for females, the presence of any males, and this was either in a mixed sex environment or the opposite sex environment, had significantly shorter lifespans. But there was also a surprisingly negative effect of being with the the same sex. So being only with females also significantly reduced female lifespan, which is quite interesting because I think a lot of people don't really focus on the costs of female-female competition. And for males, we found that exposure to same-sex rivals had no effect on male lifespan, but exposure to females significantly shortened it. But the exposure to both rivals and females had the strongest effect on male lifespan, which is interesting because it should suggest that both mating and male-male competition for mates is really costly for males. When we looked at how mortality changes in response to the social environment, we found that exposure to males accelerated senescence in females, which is the top plot, and it's the green and the dark green curves. And so these are the social environments where males were present. And it didn't really matter at what level males were present, just any male presence really was bad for females. And for males, the only environment in which there was significantly accelerated rate of aging was the one where males were with both females and males, and that is the dark green curve. And when we look at age-specific reproduction, so the number of offspring that was produced during those standardized weekly mating assays, we found that there was overall no effect of social treatment on offspring number, and there wasn't an interaction between the number of offspring produced and age, but only for males. So what we found basically was that males don't really show significant reproductive senescence across any of the social environments. They seem to be able to maintain their ability to produce offspring until they die. But there was also a bit of significant zero inflation in the mixed sex and opposite sex groups. But in contrast, females, regardless of their social treatment, they produced significantly fewer offspring as they aged. That's kind of expected. But I think the extent to which that happened, particularly for the females that were mostly kept alone or with only the same sex, is quite surprising, which kind of suggests that there is widespread reproductive senescence across all of these environments, and that just mating is just so bad for females, just really drops off so quickly what they can produce. Okay, but also our experimental design had some flies that were with the opposite sex for a period of time before the standardized mating assay. And so obviously for males, that means that they had access to either 2 or 3 extra females that the males in the alone and the same-sex treatments didn't have. And so the offspring that they were producing during this time could obviously account for some of these differences. And so to consider this, we decided to count the offspring from the social treatment groups during weeks 1, week 3, week 6, 7, and 10, which was kind of to get a spread of like early, mid, and late life periods of their life, and add that offspring count to the reproduction counts and see if that changes things. And so when we include those offspring counts, which are indicated by the black dotted lines, we find that, yes, males do have more offspring in the environments in which they had females available. So these lines are the cumulative total number of offspring for each individual in our study. The blue is obviously males and the red is females. But you can see that when males had 2 females present, and that's in the mixed sex environment, they had almost double the number of offspring compared to alone or same-sex males. And when males had 3 females present in the environment, they had almost triple. So the number of females does really affect the number of offspring that males can have. But surprisingly, we don't really see any difference for females. They don't really have a fitness benefit to having extra males in their environment. And when they get into the opposite sex environments, so they're just with males, you can kind of see that they don't really have quite as many offspring as the other social groups, which is really cool. And then when we look at the time it takes males and females to reach about 80% of their total fitness, which is almost their maximum, but not quite, over time, we can see that across all the social treatments, males were either the same age or even older than females when they achieved 80% of their total fitness. So the alone and same-sex males were about 7 weeks old when they achieved this number, but the mixed and opposite-sex males achieved this goal much more rapidly at around 3 to 4 weeks of age. But for females, they actually reached 80% of their total fitness much earlier. In almost all social environments, they were much faster than males. So these findings kind of suggest that males have more plastic life history strategies in response to their social environment. So they can live fast and die young, or they can live slow and die old. But females seem to have consistently adopted a live fast, die young strategy that did not change with the social environment or additional mating opportunities. So we know that males can influence the costs of reproduction for females and that these interactions can negatively impact female lifespan. But our results go a little bit further and kind of show that male interactions can accelerate female aging, such that females that have consistent mating opportunities throughout their life, even when they are alone most of the time, can experience really rapid actuarial and reproductive senescence. And I think that's really interesting because it highlights just how costly reproduction is for females. I mean, it's like an expected result, but just the extent to which that happened for females in our study is really exciting and quite interesting. And so this is why understanding the dynamics of male-female interactions throughout their life is a really important step to better understand some sex differences in lifespan and aging. So with that, I'll just say thank you to everyone involved on this project. So Tracy, Alex, and Amanda, and also Jess and Emily Churchill. Thank you very much.
All right, thank you everybody for coming today. I'm going to talk about some data that was collected by my graduate student, Brooke Peckinpah. So if I say we, it actually basically means her. So as many of you know, pre-mating traits are wildly diverse. We see them out in the wild, and there's massive amounts of diversity as a repeated pattern. And especially in male traits, that diversity is incredibly apparent, and the prevailing explanation for that is because of sexual selection. So it raises the question among some of us that sexual selection doesn't stop after mating stops. So are those same patterns obvious in traits that arise after mating has occurred? So some studies that have focused on this have shown really beautiful, extravagant diversity specifically in male traits, especially in sperm and sperm-related traits, which in and of itself also raises the question about whether this diversity that we might see is primarily in male traits, and then also whether the evolutionary drivers are the same on these post-mating traits as they are on pre-mating traits. So we were very interested in addressing these questions by thinking about the pattern and rate of trait evolution in these post-mating traits, both males and females, and addressing this in a macroevolutionary context using phylogenetic comparative methods. The reason for that is that these methods are really powerful. They allow you to estimate and compare evolutionary rates in individual traits by knowing tip states, but they also allow you to test causal relationships, for example, thinking about correlations between traits, while you account for a shared evolutionary history that might be misleading unless you considered the phylogenetic context. So the comparative framework that we investigated this in was in Drosophila. So we just heard a beautiful talk about Drosophila. And we were able to do that by essentially leveraging our data on this very nice phylogenetic framework that has been generated by Suvorov and colleagues, where we could use their topology and their node dates in order to generate the phylogeny for which we could assess trait evolution. And then on top of this phylogeny, we measured a bunch of male and female traits, mostly focused on reproductive variation, directly in 15 species, and then we augmented that with data drawn from the literature for an additional 15 species. So among the traits that we measured was morphology in male, so if I'm talking about trait, some of the traits that we focused on were non-reproductive, so body size and other related traits. But the post-mating sort of sexual, internal reproductive traits we're interested in include some of the ones that are shown here. And the ones that I'll probably focus most on today are testes length, which is an obvious reproductive trait that many people have studied, as well as sperm length. In females, we focused on similar kinds of traits, so some non-reproductive body morphological traits, in addition to internal sexual characters, including 2 sperm storage organs, and the one that I'll focus on mostly today is the seminal receptacle, also sometimes called the ventral receptacle, which is the primary sperm storage organ in Drosophila. So with those traits, we could address a number of questions, including the pattern and rate of evolution. We could compare rates among different sexes, and we could look at the relationship between those traits. We also measured a set of developmental or behavioral traits. I'm just going to focus on one, and that's female remating rate today. And the reason for that is, as many of you know, remating rate is a common proxy for post-mating sexual selection. So the reason for that, as many of you know, is if females remate, it means that more than one male sperm and ejaculate is in her reproductive tract at the same time. That can amplify the opportunity for post-mating sexual selection, and the idea is that that may lead to more post-mating evolution. So by adding that trait, we could also look at the relationship between post-mating morphological evolution and remating rate. Okay, so what did we find? Well, we looked at post-mating traits and we found out that they were very diverse. So here's just some pictures that show you some of these post-mating reproductive morphologies. Here they are in males. Here's some example in males. In particular, testes length, and there's about an 1.5 magnitude of difference in some of these traits. So you get all the way down from a mingy little testes that's less than 1 millimeter all the way through to more than 2.5 centimeter testes, which some of you have already seen in your intro bio in these groups that have sperm gigantism. And just to show you my favorite one, that's actually from Drosophila novomexicana, and the cool thing about these ones is that when you dissect the sperm out of the bursa, if they've mated, they're like these little slinkies. It's super cool. Okay, so female reproductive trait variation is even more extravagant. So I'm showing here these white boxes that are overlaid on top of that seminal receptacle, and you can see that in some of them it's really tiny, less than 0.5 millimeter, and in some it's massive, it's 3.5 centimeters basically, and it's like a big intestine, right? So some of you will have seen these before, but like the first time you see them, they're sort of shockingly amazing. Okay, so we can use these phylogenetic comparative methods to essentially reconstruct evolution over time, reconstruct the ancestral states for each of these traits and show their evolution across the phylogeny, which is what's being shown here with these sort of shaded comparisons. We can also use phylogenetic comparative methods to estimate the rates of these traits. So here's the estimated rate from a comparative framework for the male and the female traits, and these are actually evolving relatively rapidly, especially in comparison to some of those other body morphology traits that we measured. However, it is definitely true that there is no evidence, in fact we can show that sort of visually here, but also using comparisons of models that include single versus more than one rate, that we have no consistent evidence that male traits are evolving faster or are more exaggerated than female traits. In fact, what we really see is that there's really strong covariance between males and females. So you can show that here by looking at the correlation between different kinds of traits. At the bottom here we have testis length, and at the side we have seminal receptacle length, and you can see that they're very tightly correlated. And actually you can also see that when you take into account phylogeny, there's still a very tight correlation. So that means that there's evidence for coevolution even once shared history is taken into account. This is with phylogenetic independent contrast, but you can also do it with other kinds of models like phylogenetic generalized least squares. We show a consistent pattern that survives phylogenetic correction. What about remating rate? Well, we also find a lot of interesting variation in remating rate. Brooke spent about 1.5 years quantifying the age to reproductive maturity and quantifying the average interval between the first and the second mating. It's quite a lot of work to do that. Here's some of the data. And as other people have also shown, there's a lot of variation in this trait. So I've just boxed here, there's a bunch of species that the females will remate within 1 day, and then there's other species where it takes at least 10 days and sometimes up to more than 20 days for that female to remate, depending upon the species. So we can look at the relationship between this trait, as I mentioned, and reproductive morphologies in both males and females. And if you do that, this is what it kind of looks like. And if you squint hard, you can kind of see that there is a relationship between remating rate on the X and measures of morphological trait variation in these post-mating reproductive traits. It's fairly weakly correlated for both males and females, males at the top and females at the bottom. However, so that's consistent with this proposal that, that might impose stronger selection, but not after accounting for phylogeny. So if we do phylogenetic independent contrasts, that relationship evaporates, alas. It also evaporates in the same way when you do all sorts of other kinds of comparisons. So here's some data from phylogenetic generalized least squares, a bunch of different traits that we measured, and there is no relationship once phylogeny is accounted for between remating rate and these morphological traits. So one concern might be, well, there's only 15, I didn't measure these traits, but like goodness, there's only 15 species. Maybe there isn't enough power. So the other thing that we did was source additional variation in the literature. As many of you know, people have studied remating rate for a long time, and most of the data in the literature is collected in slightly different ways, but what you can do is pull out discrete categories of infrequent maters and rapid rematers, where the rapid rematers are females that remate more than once in a day. So we pulled out that data, and that gave us like about twice the data set size. And so here's those data with the trait evolution estimated in a slightly different way because it's a discrete trait. If you just visually look at that, you can sort of see that the patterns are quite similar in terms of how that trait is reconstructed on the phylogeny, but I can also tell you that if you do phylogenetic tests using these comparative methods, again, they're slightly different because they're discrete traits. We find no relationship between reproductive morphologies and remating rate, and that's with these threshold models, and that's with multivariate Brownian motion models, and that's with whatever model you want to fit. So actually, maybe the most striking way of seeing this is to sort of look at this relationship in phylomorphospace. That's what's being shown here. On the x-axis is sperm length, and on the y-axis is actually SR length, which I've lost somehow. And you can see there's two different categories being shown here, rapid rematers and infrequent rematers, and that correlation is identical. In fact, it's like just slightly stronger for infrequent rematers than rapid rematers. Okay, so what did we show? Okay, what did I show? We can characterize the rate and pattern of trait evolution. We don't see any evidence that male traits are evolving faster than female traits. We do see really strong patterns of correlated evolution, but we do not see any association between that and remating rate. So what do these things mean? Well, patterns of strong male-female covariation are not like exclusive to us. Lots of people have shown this pattern before in similar kinds of data sets. So here's two of them, one from Scott Pitnick's group a long time ago, one of many of those studies, and one from Luke Holman and Rhonda Snook. And you can see that there's a strong correlation. These are measured in slightly different ways, but like the pattern is very similar. So this usually indicates really strong male-female post-mating co-evolution. So that's clearly something that's going on in this system. I don't really have time to talk about it, but one of the things that Scott Pitnick first pointed out was that these relationships show positive allometry. So that means that the female trait is evolving slightly more rapidly than the males, and that's what we see too, and we think that that suggests something about the females going first, which I'm happy to talk about later. What about remating rate? Do I think it has nothing to do with post-mating reproductive evolution? I don't think that, but it clearly is a little bit complicated. So I think part of the complication is that it is both a driver and a product of post-mating sexual selection. And so I think it's doing something much more dynamic that is uncoupled to this morphological evolution that we've been looking at in this macroevolutionary context. We see that it's more labile than those morphological traits. We know that it co-evolves with mating duration and presumably with other sexual traits, which we're interested in looking at further. But what we do know is that there's no evidence that it is a driver of this sort of sperm gigantism or any of these sort of macro internal traits that we see that are beautifully diverse in Drosophila. In fact, I think it's probably a response to that. And it's hard to see, but you can see that the transition to exaggerated morphologies actually occurs earlier than the first transition to rapid remating on this phylogeny. Okay, with that, I will acknowledge that the person who really did most of this work is Brooke. Here she is holding a snake. I don't really know why, but she is. We also had a lot of really generous people share fly lines with us because, sourcing a bunch of fly lines, it's actually super cool to have like dozens of flies in your lab and be able to do that. And some funding from a training grant from the NIH. And with that, I'm happy to take questions if I have time. If there are questions. Yeah.
Thanks for the talk. You mentioned there at the end, but did you find a correlation between mating duration and remating rate?
Yes.
Thinking maybe that you didn't measure the amount of sperm between the lineages.
It's actually really hard to measure the amount of sperm in like multiple different, but we know that mating duration is negatively correlated. So if you have a short mating copulation duration, you have a rapid remating, presumably because of sperm limitation and sperm replenishment. So there are all sorts of interesting, I would say, mating strategies that are clearly evolving there, but they're just not really shaping this other sort of like macromorphological variation. Thank you for the question. And now I shall depart. Thank you.
Hi, everyone. Thanks for sticking around. My name's Dan Powell, and I want to talk to you about some work we've been doing in the Schumer lab lately, looking at how complex structural variation and behavioral interactions underpin a balanced sexual mimicry polymorphism in Xiphophorus birchmani, the sheep's head swordtail. But first, I want to start with an acknowledgement. Everyone you see here has contributed to this project in one way or another, but I want to particularly shout out Tris Dodge, an excellent graduate student in Molly Schumer's lab whose keen interest in structural variation has driven a lot of this work. So how phenotypic diversity arises and is maintained within populations is a classic puzzle in evolutionary biology, because we know that balanced polymorphisms segregate within sexes of many species. Here are just a few examples. But I want to talk about sexual mimicry in particular. This is when individuals of one sex imitate the phenotypes of another, because this is a widespread type of polymorphism that has evolved across the metazoan tree of life. This can take many forms, including morphology, coloration, chemical cues, behavioral phenotypes, and even delayed maturity. And often a whole suite of these traits are co-inherited. Now, sexual mimicry polymorphisms often, but not always, evolve as alternative reproductive tactics. In this context, it's kind of a a coercive mating strategy usually, as in the case of the bluegill sunfish, where satellite males, these female-looking satellite males, gain matings by invading the territories of more colorful parental males. But like I said, it's not always about reproduction. Sometimes it has to do with reducing social harassment, as happens in some birds. And in the case of garter snakes, sometimes it's males will mimic females in order to raise their core body temperature after hibernation. These things are often hypothesized to be under balance. Oh, did I not put that up? Yeah, okay. Often hypothesized to be under balancing selection. But there are very few examples of these sexual mimicry polymorphisms where we know both the causal genes and the selection mechanisms that might be underpinning them. So I want to tell you about our attempts to do this in a fish that I love, Xiphophorus birchman eye. Just to orient you to the system, this is what a male looks like. He's big, showy, and beautiful. This is what females look like. Also beautiful, maybe not quite as showy. You can see that there is clearly dimorphism between males and females. But a subset of males look like this, where they've got a pigmentation, a melanophore-based pigmentation spot on their flank. We call this the false gravid spot. And we call it the false gravid spot because it looks suspiciously like the true gravid spot on the females. This is a sign of sexual maturity in females. Now, I'm not trying to convince you that this male convinces anyone that he's a female, because he's also big and showy. But when we look at smaller, less ornamented, younger males, the story's a little different. These are both males, one without the spot and one with. But if I put that one with the spot next to a female, you'll see that it might actually be a pretty convincing sexual mimicry polymorphism. So, with all that in mind, I want to address two main questions. What are the genetic and genomic architecture underlying the false gravid spot polymorphism? And what mechanisms might contribute to the long-term maintenance of such a polymorphism? Let's start with the genetics and genomics. Okay, so the first thing we did was the GWAS. We sequenced a bunch of fish, some with the spot and some without, all males, and we recovered one single peak on chromosome 2. But it's a very strong peak. It explains almost all the variation in this trait. And it's quite narrow. It's only about 17 kb wide. It falls in an intergenic region just upstream of a gene called KIT ligand A, which is a great candidate for a pigmentation polymorphism because it's known, it's KIT ligand A, see? Because it's known to control coat color in mice, blonde hair in humans, flank coloration in stickleback fish, as well as head coloration in betta fish. And importantly, usually this variation is regulatory in these systems. And remember, my peak fell in an intergenic region. So, zooming in on this peak, I wanted to align some haplotypes, one of a male without the spot and one with the spot. We had some long-read sequencing to do this, but I want to orient you to what you might see when you do these alignments. If they're syntenic, you expect a one-to-one relationship like this, but if something else is going on, things get funky. This is what an inversion would look like, or a duplication. So, spoiler, or not, I guess I'm just telling you, it looks really funky in this region. And it involves several variants, including an inversion, 2 duplications, and an insertion, and this is in just the first alignment we did. So given that this change was in a non-coding region, we wanted to do some expression data analysis, and we found that not only is this allele differentially expressed in the testes and not in many of the other tissues, but if we looked at the 300 kb region around this peak, it's the KIT ligand A is the only gene that's differentially expressed between fish with and without the spot. We also did some allele-specific expression with an F1 cross and found that the false gravid spot allele is in fact upregulated. I went through that really fast. We did a lot of expression data analyses, and I'm happy to talk more about it with anyone who's interested. But we wanted to see if we could see signals of balancing selection in the genome. We have enough data that we could do some demographic history inference at 3 different populations of this fish. And so we did some neutral evolution simulations and found that it's very unlikely for this polymorphism to be maintained without some sort of selection maintaining it. In our most bottlenecked population, only 0.23% of the simulations maintain the polymorphism. So then we looked at some PopGen statistics. Nucleotide diversity, it falls in this region falls in the top 0.24% across the entire chromosome number 2. Tegemus D is not as stark, but it's still in the top 10%, actually in the top 7% of the tegemus D across that chromosome. And then we also looked at the non-central deviation statistic, and it is in the top, or the bottom 1%. So these are all signs that this very well may be under long-term balancing selection. So given that we have the signal that it may be under the balancing selection, we wanted to look at what mechanisms might be maintaining this. So the first thing we did was a developmental analysis where we tracked the development of the false gravity spot over time in 40 individuals, and we found, here's where it shows up, we found that it always shows up long before sexual maturity. And it shows up also before any of these secondary sexual traits that make those males so big and beautiful. So we thought that, well, maybe this might be a way to avoid social harassment, but there are a lot of things that might be maintaining this. And so we came up with several hypotheses that we wanted to test. One is there might be some sort of pleiotropy going on, could be beneficial, could be costly, but we know the KIT ligand does have behavioral effects in some other species. Then we thought, well, maybe there's a male-male interaction component to this. For example, if males with false gravity spot receive less aggression from larger, more aggressive males, then that could be beneficial, but if they receive more attention because they're actually fooling them into thinking they're females, that could be costly. Or there may be a mate choice component to this. Do females prefer males with false gravity spot or males without? So first, to look at the pleiotropy question, we only measured one behavior, but that was boldness, where we measured the male's willingness to explore an uncomfortable environment, that is the white background versus the black background. And what we found is no difference between false gravity spot males and not the non-false gravity spot males. Lost some of my labeling there. But yellow, actually none of those are the means. They're actually supposed to be data on there too. Does it look the same for you as it does? Yeah, it does. Okay. Okay, well, those are the means, and there's no difference between the two. Purple, false gravity spot, yellow, non-false gravity spot. So then we looked at male-male aggression. We set up these open field trials where we put a large focal male in with a female, let him get comfortable, feel like it's his home territory, and then we introduced size-matched small males, one with false gravity spot and one without, and we measured both aggressive as well as amorous interactions. And what we found on the aggression front, again, you can only see the means, but the false gravity spot males received a lot less aggression than those that did not have the false gravity spot. This is going slow. Okay. Yeah, all right. They also received a lot more amorous attention than the males without false gravity spot. These are courtship interactions as well as attempted fertilizations. There was a lot of same-sex behavior towards the males that had this female mimicry trait. Okay, so that's male-male interactions. We wanted to know how the females felt about this, so we made some animations so we could control for behavior, one male with a false gravity spot, one without. We put them on a computer monitor on either side of a trial lane and asked the female who she wanted to spend the most time with. Turns out females spend about 40% more time with males without the false gravity spot over males with the false gravity spot. So these are our first attempts at trying to understand the mechanisms underlying the maintenance of this trait. But if the slide will ever switch, I will summarize. All right, this is not fun. Okay, so we found three possible mechanisms that might be affecting the balance of this polymorphism. We found reduced aggression, we found increased amorous attention, which is probably costly, although we could make arguments either way for that one. But we also found that from a mate choice perspective, it's not great to have that spot. So these things are opposing forces, right? And this could be maintaining the balanced polymorphism. There are other things that could be maintaining it as well, and it may be a case of frequency-dependent selection or something like that going on. So we have a lot of future directions to go, but this is where we are now. So I'd like to summarize for you what I showed you. There's a complex structural genomic variant associated with the gene KIT ligand A that underlies the expression of false gravid spot. The haplotype drives allele-specific expression, and patterns of genomic diversity indicate balancing selection could be acting on this phenotype. There are opposing selective forces acting on individuals with the phenotype: reduced aggression, increased same-sex attention, and negative female mate preference interactions, which could be maintaining this balanced sexual mimicry polymorphism. Like I said, there are a lot of future directions. We want to disentangle the context of dependence of selection acting on the balanced mimicry polymorphism. We need to uncover further selective mechanisms acting on the phenotype in nature. We did do some field observations, but those data are very messy, and we need to do a lot more observations. But also, what I didn't tell you is this trait is not only polymorphic within the species, but also across the genus. In fact, across up to 6 million generations. And so it has been maintained for a very long time, and we really want to try to understand that deeper pattern. And further, we want to know what the role of integration in the distribution of this trait is, because there is some evidence that this has integressed from other species across deep nodes in the tree. So, shameless plug time. If these sorts of things interest you, or you're interested in the evolution of reproductive barriers and the causes and consequences of their breakdown, and you want to use genomics or behavioral ecology, work with natural populations or lab crosses, and you like beautiful places and awesome fish, my lab is starting this fall at LSU, and I'm recruiting students, so please hit me up.